Thank you very much, Brian, for your excessively kind introduction, and thanks to the Society of Fellows here for inviting me back to Scripps. It's always fun to come back and talk to people here, so we've had a great day. It was supposed to be a great 2 days, but one day got cut short, so anyway, I'm glad that I finally made it here, and I'd like to tell you about some of the work that we've been doing over the last decade or so, trying to build structures that look like what we think the first cells on Earth might have looked like. And so that involves basically trying to understand how you take a bunch of molecules made by prebiotic chemistry and get them to come together and start acting like a cell that can, most importantly, evolve in the Darwinian sense. So these kinds of questions, of course, have been around for a long time, for millennia, I suppose. But I think the whole field has really come into greater public attention, not because of what we in the field of origins have been doing, but more recently because what our astronomer friends have been doing in finding evidence for solar systems like this. So a lot of this work comes out of the Kepler Space Telescope, which has discovered many solar systems with planets orbiting around other stars, some quite similar to our sun, as in this case of Kepler-62. So Kepler-62 has 3 inner planets that are way too hot for liquid water or for life. But interestingly, it has 2 outer planets that we think are in the habitable zone where there could be liquid water. The inner one is probably a little bit on the warm side, but could still potentially have liquid water on the surface. And the outer one is probably a little on the cool side. It may be more like Mars, although its mass is much greater, so it could also potentially have liquid water environments. So from observations such as this, the current extrapolations from the Kepler data are that there may be hundreds of millions of Earth-like planets orbiting more or less sun-like stars in our galaxy alone. And of course, in the universe as a whole, there are, I forget, on the order of 10 to the 12th galaxies. So we know there are many places out there that could support life. Right? But what we don't know is whether any of them actually do. And so that comes really down to this question. Is it easy for life to emerge from the chemistry that occurs on young planets? Or is there some step or series of steps that are hard and maybe in combination are so difficult that life might be an incredibly rare phenomenon in the universe? So the extreme possibility is that it's so hard for life to get started that we're the only place in the universe that has life. The other possibility is that there's a simple, straightforward pathway composed of easy steps, and maybe all of these planets have life on them. And it's kind of interesting, we don't know, and yet people tend to have passionate opinions one way or the other, that life is just a universal attribute of the universe and it's found everywhere, or it's so special that it's only here. So that's basically what drove me to start trying to look at this and just do some simple lab experiments and see if we can contribute to this effort of defining a pathway. So I'm not particularly interested in where we draw the borderline, what we call life or not life. But what I would like to understand is this pathway that goes from planet formation to simple chemistry to more complex chemistry, self-assembly processes that lead to the formation of cell-like structures, and then the next steps where these structures can start to grow and divide, transmit information from generation to generation, and therefore gain the capacity to exhibit Darwinian evolution. I think once you get to that point, the pathway, the next steps to much more complicated biology and ultimately to us are easier to understand. So it's those missing links in that pathway that we're trying to fill in. Okay. So why has this been so hard to get at. And I think at least historically, a big part of the conceptual barrier has been that modern life is so complicated, right? At every level. I mean, at the structural level, cells are complicated. Underlying that, you have this incredibly complicated metabolic chemistry, thousands of catalysts, thousands of chemical reactions that all have to be properly accelerated and coordinated. And underneath that, of course, you have this flow of information in cells, which by itself is not a problem, but the issue is that every part of this flow depends on every other part. So you can't make DNA without RNA and proteins and metabolites. You can't make proteins without DNA and RNA and amino acids and all the whole complexity of the translational system. And you can't make this molecule in the middle without stuff above it and below it. So how could such a self-referential system emerge spontaneously, right? And so this was such a difficult issue that it led to all kinds of bizarre theories of the origin of life for decades, right? Until the discovery that RNA molecules could act as catalysts, right? And so that immediately led to much simpler molecules. And of course, there were a few really bright people, like Francis Crick, Leslie Orgel, who thought that, you know, maybe this is how we got started. They published papers on this in the 60s, but it really wasn't taken seriously until Tom Cech and Sid Altman discovered specific biological examples of RNA catalysts or ribozymes. Okay. So with that discovery, it's possible to imagine RNA catalyzing its own replication, and lots of interesting work has been done, still going on here, for example, and I think we're getting closer to seeing this, but we're not quite there yet. You could also imagine RNA carrying out metabolic reactions, synthesizing the components of primitive cells, things that would help the cell grow and divide. But we need examples, right? We'd like to actually see that happen in laboratory experiments. And so this kind of hypothesis of an early stage in the evolution of life, where we have essentially one informational biomolecule, a genetic polymer that can also act as a functional polymer, is generally called the RNA world hypothesis. And I think the real questions in the origin of life are, how do you get from the chemistry of a young planet to the RNA world? That's where we have a lot of gaps in our understanding. Okay. So there were a lot of people working on the earlier stages leading up to this. You know, how do we make nucleotides? How do we make lipids? How do we make amino acids and peptides? What we've focused on is what happens next, right? So we're assuming that all our chemistry friends will solve those problems, give us the biological building blocks, and we'd like to know, then what? You know, how do they assemble into structures that look like primitive cells, protocells? A cell membrane, presumably very different from modern membranes, so it has to be a membrane that's not a barrier, right? Because remember, at the beginning, we didn't have sophisticated transport machinery. So it has to be a permeable enough membrane that nutrients can come in from the environment. We need some kind of genetic material, and for reasons I'll explain, we think now this is most likely to be either RNA or something like RNA, although there may be people here who have different opinions, that's okay. We, in any case, would like to understand how structures like this can assemble. But again, I think the assembly of structures like that is the easy part. And the harder and more interesting part is, how do they grow and divide, right? What physical and chemical processes, what environmental fluctuations could drive the growth of, first of all, the membrane, and cause a cell after growth to divide into daughter cells before there was any evolved cellular machinery to do that. And then at the same time, there has to be chemistry that can copy useful sequences, physical processes that can get the strands apart, so you can do that copying again, distribute these replicated molecules into daughter cells, and all of that has to be spontaneous, environmentally driven chemistry, and all of this has to be mutually compatible. So that's what we've been trying to do, is to actually, in lab experiments, find ways of generating structures like that and working out the physics and chemistry that will give us this cycle of growth and division. And we think that if that could be done, we would essentially have a very primitive cell cycle. And by exploring larger and larger numbers of sequences, eventually, I think, some sequence would turn out to do something useful for its host cell. At which point, it would have an advantage, either more efficient replication, or greater stability, or something, some kind of advantage that would let it start to increase in abundance in its population. And that would be the beginning of Darwinian evolution. That's what we'd really like to ultimately see in the lab. Okay. So one important point is that to have these kinds of physical and chemical processes going on, that this has to be driven by a flow of energy and materials from the environment. Okay, so these are far from equilibrium systems. We have to think about ways to bring energy into the system. What's the source of chemical energy? What kinds of mechanical processes could lead to division? Do we need thermal cycling for strand separation? There are many ways to bring energy into the system to drive this process. And of course, we also need to bring materials in. We need to have a flux of biological building blocks into the system so that growth and division can occur. Okay, so I'm going to talk a little bit first about the membrane side of things, and then I'll talk about the nucleic acid side of things, and then a more recent direction for us is thinking about the way that simple abiotically formed peptides could play useful roles, either by interacting with the membrane or by interacting with nucleic acids. So we really need to think about these three components, at least, in order to build a fully functional system. Okay, so I mentioned the kinds of membranes that we're thinking about for protocells are very different from modern membranes, which have complex building blocks like phospholipids, sphingolipids, sterols, and so on. We're thinking about much simpler systems based on, for example, fatty acids like the ones shown here, related molecules, so simple esters of the fatty acids, glycerol esters, fatty alcohols, polycyclic aromatic hydrocarbons can integrate into the membrane and possibly provide some stabilization functions. But even just fatty acids, for example, just oleic acid, if you just shake it up in water at the right pH and concentration, it'll self-assemble into beautiful vesicular structures. They have bilayer membranes. And so they're a very spontaneously self-assembling system. And the membranes that are formed have actually amazingly interesting properties. And so I think some of those properties are demonstrated on this video animation that was made by Jana De Wasso when she was in the lab. And the point here is that these systems are amazingly dynamic. The flip-flop is on the millisecond time scale, and these molecules exchange in and out of the membrane, and therefore between vesicles, also very rapidly in the second time scale. We think also that there are transient defects or complexes that form with polar molecules that allow even nucleotides to cross these membranes spontaneously. So you don't need any advanced machinery to get building blocks into the cell. On the other hand, they will trap oligonucleotides inside permanently, so that any useful genetic information is trapped inside these vesicles. Okay. So we worked on these for about a decade using various model systems and looking at growth and division in kind of indirect ways. But then when Cheng Zhu, a really talented graduate student, joined the lab, he decided he wanted to actually see this happen. And so he worked out ways of preparing vesicles that were large enough for us to just look in the microscope And this is a field of such vesicles, about 4 microns across. And what you're seeing is a fluorescent dye that's trapped in the internal aqueous space. And so the experiment that Ting did that really changed the way we think about growth and division is to take vesicles like this, just add some more fatty acids in the form of micelles, and watch what happens as those added fatty acids get incorporated into a preformed vesicle. And we thought they would just kind of grow and maybe become a little bit elongated if the surface grew faster than the volume due to osmotic constraints, but we didn't expect the way that these things actually do, which is to sort of sprout these funny-looking filamentous tails. And this is what it looks like after about 5 minutes. After about half an hour, they've been transformed into long filamentous vesicles. So they're still topologically closed. None of the contents have leaked out. But these structures have the advantage of being very fragile. And so division into daughter cells is now much easier. It's really hard to divide a spherical vesicle, especially a swollen vesicle. It's very easy to make these divide. And so I'm just going to show you a few movies of the process. Okay, so the first one is the growth. So here you're looking at a single vesicle. Only 8 micelles have been added to a buffered solution. That makes the micelles thermodynamically unstable, and the molecules gradually integrate into the preformed membrane, which grows into this filamentous structure. So I like the movie so much, I'm going to play it again. So it kind of, in a way, it looks very biological, right? There's a lot of motion and shapes are changing. But remember, it's just soap and water. That's all it is. Okay. So once these things form, once you have filaments, then the question is, well, what does it look like when you want to divide them? Okay, so we have many ways of making growth occur now. I'll show another one. We have 2 pathways that lead to division. So one is just simple mechanical energy, just shear forces in the environment. So a puff of air on the microscope slide is enough to trigger the processes you see here. So there's this purling instability which occurs, and in between these beads seems to be a kind of weak junction, and mild shear forces will cause the filament to snap. And then if you just let them sit, they'll round up. You have smaller spherical vesicles, which you can then grow into new filaments and repeat that cycle ad nauseum. Another way of doing it is through a more complicated photochemically initiated process, which is shown here, and it's kind of dramatic in a different way. So much more pronounced purling instability, which is triggered by changes in surface tension. And then the individual beads gradually separate from each other and diffuse apart. So this is a way of getting many small progeny from one parental vesicle. And again, you can get these smaller vesicles to grow and go through more cycles of division. Okay. All right, so like I said, we have several different pathways that lead to growth, a couple of ways of leading to division, and we can make this into a cycle. So it's always very satisfying in this line of work when you have more than one way of doing something, right? Because that means that the whole process can probably occur under more than just one restrictive environmental condition. There's lots of different ways this can happen. Okay. So the growth pathway that I showed you would be dependent on a rapid influx of new nutrients, new fatty acids, right? You can imagine scenarios where that could occur, but it would be nice, we thought, to have different pathways, maybe where growth is more of a competitive process. What if one protocell can steal molecules from its neighbors and use those to grow? And we've, over the years, worked out a pathway that works in that way. And this work started from experiments done by Irene Chen. She was a biophysics graduate student in the lab several years ago. What Irene found is that if you make osmotically swollen vesicles, Okay, so swollen because they have a lot of stuff inside them. The stuff can be anything from sugars to RNA molecules. These swollen vesicles are perfectly stable by themselves. Okay? But as soon as you mix them with relaxed vesicles that have less of whatever this stuff is inside, because of the dynamic exchange of molecules between membranes, the swollen ones now have a relaxation pathway open to them, so they can actually grow at the expense of neighboring vesicles. So it's a competitive growth process. So we were really excited about this at the time because of the aspect of competition. It's a physical mechanism for competition between these simple protocells. But the problem was we never could figure out a way of making these growing vesicles divide. And so the answer to that came several years later from yet another brilliant graduate student who worked on these membrane-related problems, and that's Itai Guten, who's now a Miller Fellow at Berkeley. And Itai wasn't actually initially trying to solve that problem. He was wondering about the transition pathway from primitive membranes based on single-chain amphiphiles, like fatty acids, to modern membranes based on two-chain amphiphiles, like phospholipids, or molecules with much higher buried surface area. So these membranes are really good as barriers. So they're very good for highly evolved cells that have the machinery to control transport across the membrane. And the question is, how could you get from this primitive state to the modern state? And the only reasonable pathway we could think of is some intermediate where you have mostly fatty acids and maybe a little bit of phospholipid doped in there. But the question is, well, what would be the selective advantage? I mean, if a catalyst arose in the protocell that could make phospholipids, you could make this kind of structure. But what good would that be? And we couldn't figure it out. So Itai just decided to do the experiment. So he made vesicles with these kinds of mixed membranes and just started looking at their properties. And what he found was pretty amazing, which is that if you make membranes that have a little bit of phospholipid in them, they will absorb fatty acids from neighboring vesicles that don't have phospholipids. And so most of his thesis was trying to figure out why this happens. In a nutshell, there's a local ordering in the membrane around phospholipids, which slows down the off rate, slows down the dissociation of fatty acids from the membrane. The on rate is the same, so the net effect is that phospholipids confer this growth phenotype, and they grow by stealing molecules from their neighboring vesicles. And you can see that down here over the course of a few minutes. The spherical vesicle is growing into one of these long filamentous structures again. And so here we have growth into a filament based on competition. So you don't need any influx of new materials in whatever environment these protocells are living in. It would be driven by the development of an internal catalyst that could synthesize phospholipids. Then more recently, yet another graduate student in the lab, Kate Adamala, showed that you could do the same thing with hydrophobic peptides that localize to the membrane. So any internal catalyst that could drive the synthesis of hydrophobic peptides would again have a growth advantage and could grow to make filaments, and assuming that division is environmentally driven, you would have a faster cell cycle. Okay. All right, so the basic lessons that we drew from these experiments is that making either phospholipids or hydrophobic peptides would give you this growth advantage. You'd have a huge selective advantage in favor of generating these catalysts by an internal coded process. That would cause a whole cascade of secondary selective pressures, ranging from pressures for more advanced replication machinery to transmit that information, but also an evolutionary arms race, right? Because now as soon as the cell that can make a little phospholipid takes over the population, then that mechanism doesn't work anymore, because now everybody's got phospholipid. But it turns out, experimentally, whoever makes more phospholipid wins. Right? So if you make a bit more phospholipid than your neighbors, you can still grow by essentially by eating them. And so that would drive a gradual increase, a transformation of the structure of the membrane from single chain to two chain amphiphils. And the trouble is that once you get up to over 50% phospholipids or other amphiphils in the membrane, you start to really change the biophysical properties of the membrane. The permeability starts to drop, and now that would allow a whole new set of things to happen. So it would become, for the first time, advantageous to start making metabolites in the cell, because they're not just going to leak out and feed your neighbors, right? You get to keep them for yourself. It would also, of course, be, for the first time, useful to start evolving transport machinery to import molecules that are good or to export molecules that are bad. Okay, so we think that these kinds of membrane biophysical considerations might have been really important drivers of early evolutionary processes. Okay, so in order for any of that to happen, of course, we need to be able to transmit coded information. We need to be able to have molecules that can make catalysts, and so we need to be able to have some biopolymer that can act both as a genetic molecule and as a functional molecule. We know, of course, that RNA can do it. We think that there are probably other things that can do it, but we don't quite know how to do that replication yet. So when we're thinking about the replication of molecules like RNA, there's a whole list of problems, which I'll go into in a minute, but I think there are sort of two ways of thinking about it. One is to use a macromolecular catalyst, for example, a ribozyme. The other is chemical replication. And I think the way I'm kind of thinking about things nowadays is that to get started, the easiest way might be some purely chemical process that could copy templates. Maybe you could even do replication cycles purely chemically. And then once you have some sequence that's actually advantageous and you want to transmit it more efficiently, now you have a selection pressure that would favor the evolution of macromolecular catalysts of replication. No matter which way you catalyze replication, there are major constraints. Okay? And some people think that, well, you know, the origin of life, you've got hundreds of millions of years, everything can go on really slowly, but if you're thinking about molecules like RNA or even DNA, that's really not true, right? So the rate of copying has to be at least faster than the rate of degradation. And if we're thinking about highly activated monomer building blocks, the rate of replication has to be on at least a comparable time scale to this, whatever the stability is of those building blocks. So it can't be infinitesimally slow. We actually need to get right going with a pretty good rate. And similarly, the fidelity of the copying process also has to be pretty good right from the start, because we need to be able to transmit enough information to generate a functional structure. We don't know exactly how much that might be, but even simple ribozymes that have reasonable rates tend to be on the order of 50 to maybe 100 nucleotides, and that means the maximum error rate that you could tolerate might be on the order of 1 or 2%. And so high compared to modern biology, but a challenge for doing this just by chemistry. Okay. So just stepping back a bit, so we have the whole problem of making these polymers and then replicating them. So there are really, I think you can divide the problems into three sort of classes of sub-problems. The first is making the monomer building blocks. So there are many people working on this, a lot of notable progress in recent years from the Sutherland lab in the UK. I think once you get activated nucleotides, getting them to polymerize into long chains is not too hard. In fact, you can take phosphodiazolides like this, which don't react if you just have a solution at room temperature. If you put them in the freezer, come back a week or a month later, you have long RNA chains. concentrate in the spaces between the ice crystals, and when they get concentrated enough, they start to polymerize. Also, Jim Ferris showed years ago that you could polymerize these on the surface of certain minerals, like montmorillonite clay, for example. Okay. So the part of the problem that we've been focusing on is trying to take these single-stranded molecules and copy the information and generate a double-stranded product. And of course, this is something that's been worked on for decades. This work, the major contributor to this whole field was the late Leslie Orgel. Many of his students and colleagues made a lot of progress. Jerry Joyce here contributed a lot of the key experiments. And I think it's fair to say that in the early days, it looked like this really might be easy, right? The initial experiments showed that you could take activated monomers and start to build up copies of certain template sequences. But then as time went on, it looked like there were more and more problems standing in the way of being able to do this efficiently enough to copy essentially arbitrary sequences and to generate a cycle of copying, strand separation, copying again, which is what we really need to get started. So, I would say somewhere around 15 years ago, maybe 20, more and more people, myself included, started to think that maybe copying RNA with just chemistry is just impossible. Maybe there's just too many barriers. And so many people, and especially Albert Eschenmoser, started to look for alternatives to RNA. The idea is maybe there's some simpler polymer that's easier to make under prebiotic conditions and easier to replicate. And so that led to a really very, very productive enterprise and basically led to the demonstration that yes, there are many alternatives to RNA that can do Watson-Crick base pairing. They make anti-parallel double helical structures. They look like decent genetic polymers. They probably even can do function. And recent work from John Chapu's lab and Phil Holliger's lab have shown that you can make functional folded structures from lots of things besides just RNA. But in recent years, after looking at that for quite a while, I at least have come back to the opinion that maybe life really did start with RNA or something close to RNA. Part of the reason is experiments that are starting to fill in some of the gaps of how you would go from cyanide to actual nucleotides, which is, there are various pathways that have been proposed that look much more efficient than what we used to know. And so that led me to sort of come back and look at the problem again. So I just want to show you another one of Janet's beautiful animations. This is the problem, right? This is what the goal. This is what we want: a single-stranded template sequence that can float around with a bunch of activated monomers that can find their complementary positions by base pairing and sort of click together to make this complementary strand. Okay. So why is this so hard? Oh, sorry. I want to show one of the reasons for thinking that it might have been easy. This is a recent experiment from my lab showing that if you have a C-rich template, you can basically copy it in a few minutes with activated G. This is stuff that was worked out in the Orgel lab decades ago. The problems are shown here. Okay, so for anything except these C-rich templates, the copying is too slow. Right? It becomes slower than the rate of degradation of the template, or especially the activated monomers. And it's not accurate enough. So recent experiments from a number of people, including Irene Chen, have shown that there's just the error frequency is very high, especially because of wobble pairing, UG and AC pairing in RNA. A special problem with RNA is making incorrect backbone connections through the 2' hydroxyl instead of the 3' hydroxyl. Right, so then you're making a heterogeneous backbone, and I think we all thought you could make folded structures if your backbone was going to be so heterogeneous and every molecule is going to be different. If you can solve those problems and make a perfect duplex, then it can be actually almost impossible to get the strands apart just by heating. And if you can get the strands apart, they come back together again, orders of magnitude more quickly than the optical kinds. So I mentioned the nucleotides. The activated nucleotides tend to hydrolyze. You probably have a really heterogeneous mixture to start with. We don't really know the right source of chemical energy to drive the activation process. We don't know what the real best leaving group would be. And a problem that was particularly interesting and kind of discouraging to me was that all this RNA chemistry requires high levels of divalent cations like magnesium. And yet, the membranes that we've been playing with and that look so interesting are totally destroyed by magnesium ions once you get above a few millimolar. So all the RNA chemistry looked incompatible with the protocell membranes. Okay, so so this is why we thought for a while that it might be impossible to just use chemistry to copy RNA. But for a few years ago, we started to come back and look at these. And I don't have time to go through all of these. I'm just going to give a couple of examples. But we've solved, at least at a proof of principle level, almost all of these. And I think the biggest remaining problem is the source of chemical energy to drive nucleotide activation. I think if we can solve that problem, or somebody here, if around can give us the right chemistry to activate nucleotides, I think we might be able to go all the way and have cycles of chemically driven RNA replication. Okay, so I'll just show you a few experiments that show you why I've become relatively optimistic here. Okay, so the first set of experiments have to do with this issue of magnesium being required for RNA chemistry and yet incompatible with fatty acid membrane structures. And so the answer, proof of principle type of answer, came from experiments done by Kate Adamala, same graduate student who showed that hydrophobic peptides could drive vesicle growth. And what Kate did was to conduct a small screen of di- and tricarboxylic acids, the kinds of molecules you would expect to coordinate magnesium. So shown here is its coordination to 6 waters. Here's coordination to citrate through 2 carboxylates and a hydroxyl, leaving 3 coordination sites open. And it turns out that citrate complex with magnesium is pretty amazing because it can protect membranes, and you can see that just visually. So here's a tube of vesicles with fluorescent dye. It's a very nice homogeneous suspension. Add magnesium, everything just immediately precipitates. There's a glop on the wall of the tube. But in the presence of citrate, the vesicles remain intact, nothing leaks out, they don't aggregate, everything's fine. Okay, so you can do that with a lot of chelates, like EDTA will do that, totally protect the membranes. But if you have EDTA around, it totally envelops the ion, and so the RNA chemistry doesn't go at all. Citrate, on the other hand, allows the RNA chemistry to proceed. And so the experiment shown here, we have the same C-rich template. We put in this activated G monomer. We measure a certain rate in the absence of any chelator, and we take about a 2-fold hit in initial rate in the presence of citrate. But basically, the chemistry proceeds almost unhindered. So I think it's interesting on several levels. So first of all, now things look more compatible. I think it's also telling us something about how the magnesium actually acts as a catalyst, and maybe it'll help us to understand how that catalytic metal ion interacts with the hydroxyl and the activated phosphate. Okay, so this let us do an experiment that we wanted to do for a long, long time, and that is to make fatty acid vesicles that have encapsulated primer template complexes. We can take these kinds of structures and add to the outside of the vesicle activated monomers. in the presence of magnesium and citrate. The monomers, the nucleotides, can spontaneously cross the membrane. So remember, these membranes are permeable enough for that to happen spontaneously. And then once they get to the inside, the primary extension reaction can occur, and we can copy the template. And so this is published, so I'm not going to dwell on this, but at least at this level, things are now compatible. And the real question for us now is, this shows that you can make the systems compatible, but are there other ways of doing it? And in particular, are there more prebiotically plausible ways of doing it? So there's really two possibilities. Either someone, like Brown maybe, will discover a good way of making lots of citrate in certain environments. That'd be cool. Or I think a more likely possibility is that maybe there's something like acidic peptides that can also coordinate metal ions that could do this. And really, the thing I really like about that idea is that if you look at the heart of cellular RNA polymerases, there's a little aspartate-rich peptide that coordinates the catalytic magnesium ion, and it just looks like a little piece of prebiotic chemistry at the heart of a modern enzyme. So it's very tantalizing. So we're trying to look at peptides as metal binders that might, initially maybe just do what citrate does, but maybe it can go beyond that, and they can actually start to contribute some catalytic function. Okay, so the next little story I want to tell you has to do with essentially the rate and the generality of the template copying process. So I mentioned the chemistry tends to be too slow, and also we really need to be able to do better than just copying C-rich templates. So the advances here came from thinking about, well, this is our kind of standard model, which is obviously based on what biology does, right? We have primers, we extend them with monomers, we copy the template. So that's a really convenient system for analytical reasons, for example. But I was thinking that maybe a more natural way to begin with, and I think this is probably the earlier Ordell lab way of thinking of it, is that, well, we don't necessarily have, predefined primers present prebiotically. Maybe the way to copy a template is to do it hierarchically. Monomers sit down, occasionally they join together, they make small oligos, the small oligos ligate together, and in that kind of hierarchical assembly process, you gradually build up a full-length copy of a template. Okay, so we decided to start testing that idea and do some experiments. And both Craig Blaine and Noam Priwes, also graduate students in the lab, have contributed a lot to these experiments. And so, as almost everything in this field, our expectations were completely wrong. And so let me show you what happened. So here's the primer extension paradigm. We have activated monomers sitting down on this template. And we are comparing that to just the ligation of two oligos. So we expected this ligation reaction to be much more efficient, because everything is more pre-organized. You have a more pre-organized double helical structure, which many experiments suggest that should contribute to a significant rate enhancement. And so we thought this should be much faster than the polymerization reaction, where you have all these really flexible building blocks, a much less well-ordered starting structure. So when we look at the rate of the reaction, we were kind of shocked when this is the ligase reaction, which is incredibly slow, and almost 100 times faster is the monomer addition reaction. Okay, so that was really kind of a shock, so what's going on here? It turns out that very similar observations were made by Ordell more than 20 years ago. And the idea is that there's probably some interaction between adjacent leaving groups that contributes to the observed rate. Okay, and so here's one of the really key experiments that Noam Priwest did. So he set up a primer extension experiment where we have a single monomer that can add to the primer, that's what we measure, and then next door we put a helper oligo that can be either just a 5' phosphate or it can also be activated. Okay? And so when the helper oligo, it has only a 5' phosphate, the reaction is very slow. If the helper oligo is activated, has this leaving group on the phosphate, the reaction is very fast, faster than we'd ever seen before. So I think that really proves that there's some critical interaction between adjacent leaving groups. And we think this explains why this 2-methyl-imidazole leaving group has been such a special case. In all of the chemistry that's been looked at over the preceding 50 years, 2-methyl-imidazole has always stood out as something that is, for totally unknown reasons, is by far better than anything else. And so we're trying to understand that now and trying to work out what the actual mechanism of this catalytic effect is. But mechanism aside, we can take advantage of this catalytic effect to help copy arbitrary sequences. And so this is just one example where here we have a template that instead of being one of these C-rich templates, we have AU, CG, and then some Cs. Okay, so this is the kind of mixed sequence template that has always been refractory to this chemical copying. And so, so the first set of lanes over here on this gel, you see what happens when you take this primer template complex and add all 4 activated nucleotides. And what happens is basically nothing. Right? There's no primer extension. But if you provide the set of activated helper oligos that will let each monomer come in turn, you can walk down the template and get through this AUCG part of the template. So we can get now over 70% of the primer extended through this really difficult template sequence. So, so it's not obviously the total answer to all of the problems, but we think this is a big step forward in generalizing the ability to copy arbitrary sequences. And so the hope is if we can continue to find improvements in the copying chemistry, we'll be able to eventually do what now can only be done with enzymes or ribozymes. Okay. All right, so one final little story having to do with another one of these roadblock problems, and that is the issue of, okay, we actually know now how to get the strands apart. Turns out if you put in 2' linkages, which we thought were a problem with RNA, the lowering of the melting temperature helps you get the strands apart much more easily. The problem is the reannealing. You know, once you cool things down again after strand separation, the strands hybridize together very quickly in seconds or less, and the copying chemistry is still taking hours. So we need to find some way of slowing down the hybridization kinetics to give us enough time to copy the templates. Okay. And so the results, the progress here has come from yet another amazing graduate student, Tony Jock, who found that basic peptides, arginine-rich peptides in particular, are really good at slowing down hybridization kinetics. And yet, amazingly, they still allow template copying to go on. Okay, so we do not understand at all how this works yet, but I'll just show you some of the data. Okay, so this is a time course of reannealing kinetics. So this is the fraction of initial RNA. This is RNA 15-mers, 2 complementary 15-mers, that is still single-stranded versus time. So if there's no peptide, everything anneals in a second. Okay. You put in, let's see, so this is a bunch of different peptides. So this is alternating glycine arginine at one, I can't see the concentration from here. 100 micromolar. You slow it down by several hundredfold. All arginine peptides are even better. Here's a temer with a carboxamide. So now we've slowed down the annealing kinetics by a few thousandfold. The magnitude of the effect is strongly dependent on the length of the RNA oligos, as shown here. So 7-mer will still anneal quickly, 10-mer is slower, 15-mer is really slow. It's also dependent, I guess not surprisingly, on the length and the concentration of the peptide. So this is, again, no peptide and then increasing concentrations. And we see a similar effect on peptide length. Okay. So you might think, and I did think, that, okay, this is great, we can slow down re-annealing, but anything that's going to prevent two complementary strands from finding each other and hybridizing together is probably going to mess up base pairing so much that primer extension is not going to work. Right? So that led to the following really kind of surprising experiment. And so it's shown schematically here. We start off with a duplex in the presence of basic peptides. We thermally denature the duplex. Because of the basic peptides, the strand re-annealing is very slow. The primer is short, however, so it can still anneal quickly. We add monomers, and then we look for primer extension. Okay? And here's basically one of Tony's sets of experiments. Okay, so no peptide and no complementary strand. This is the positive control. We see template copying. We see primer extension with time, longer extension with time. If you have no peptide, but you do have the complementary strand, then the copying chemistry is totally inhibited because you immediately reform the duplex. Right? The glycine-arginine peptide doesn't really help that much, but over here you see this oligo-arginine peptide. And now we can still see some primer extension. So the strand re-annealing has been slowed down, the primer extension is still working, and we think, or we hope, that this is maybe a road into finding conditions where we could do multiple cycles of basically thermocycling RNA replication. Okay, so summing up some of these experiments, our current thinking is that maybe the primordial genetic polymer was more or less RNA, but a more heterogeneous, a messier version. So we now like the idea that this primordial RNA has a mixture of 3' and 2' linkages in the backbone. We know this doesn't interfere with making folded structural defined structures that can be functional. We know it's helpful for lowering the melting temperature. We think that there might be an interesting roles for some modified nucleobases, especially we like 2-thiol-U because it enhances the strength of the UA base pair and also makes it easier to copy mixed sequences, and it also enhances fidelity. And we think that the primitive polymer could be even messier than that, and maybe you can have other things mixed in there. We know we can put in deoxynucleotides, for example. We can still get, we can still do selection experiments with those mixed polymers. So maybe things started off kind of messy, and then as selective pressures developed, and the replication machinery evolved, we moved to a more well-defined system closer to what we have today. Okay, so then the last thing that I want to talk about just very briefly is coming back to the physical environment. What kinds of environments on the young planet could have supported the formation and the replication of these kinds of structures? And so you need environments where you can accumulate these building blocks to high concentrations. So that's why I think this idea of the primordial soup in the ocean, I think, is totally wrong. I think you need localized environments where you can build up very high concentrations, reservoirs of the relevant organic materials. We need a source of chemical energy. So to me, the most likely source of chemical energy is atmospheric chemistry. Could be atmospheric chemistry driven by impacts or by radiation, but making those high energy carbon nitrogen compounds that I think could drive all these processes. I think we need an environment where they can give us transient exposure to high temperatures. The easiest way to separate the strands of a duplex is just to increase the temperature, melt them apart, but you don't want to stay hot. You don't want to cook your RNA because it's all going to degrade, right? So you need an environment that can give you high temperature spikes. Okay, and so we'll talk about what that could be. Well, the answer is it's basically hydrothermal systems, right? So lakes where you have hydrothermal vents, streams of hot water that can do this transient heating. And so, of course, there are still lots of environments like that on the modern Earth. This is Yellowstone in the winter. It's covered with ice, looks very placid, but underneath you have these vent structures, dozens of them, which are emitting plumes of hot water. And we think this provides exactly the kind of physical environment that could drive a primitive cell cycle. So the idea is that you have these protocells, they're sitting in a cold or moderate temperature most of the time, which is good for copying template sequences. Every now and then they get swept up in one of these plumes of hot water. They get transiently brought to a very high temperature, which gives you strand separation, increases permeability, so you have a flood, an influx of nutrients into the lumen of the protocell, but then they're essentially quenched as they flow out into the larger environment. So we think this could provide a physical environment that could drive a primitive cell cycle. And so I hope that over the coming few years we'll fill in some of the remaining gaps and actually be able to demonstrate this kind of system in the lab. Okay, so all of this work has been done by just a large number of amazingly talented students and postdocs. I've tried to mention most of them as I went along. Current lab members who've contributed a lot to this are known through us, and Tony Job and many other people in the lab, both now and previously, have really done all the work that I've talked about here. So thanks again for inviting me to come, and thanks for listening.